In this video, I'm just going to rep through bioenergetics because it's also helping me review for the final. So we start with the quickest, fastest system, the phosphocreatine. So we have PCR plus ADP yielding ATP and creatine, and the rate limiting enzyme is creatine kinase. And then we move on into glycolysis. So we start with a glucose molecule, and then hexokinase breaks it down into G6P, and then PGI will break it down into F6P, and then we have phosphofructokinase, which is going to yield F16 bisphosphate, and then aldolase splits it down the middle, and then we get PU and OOP, and then we get two pyruvates. One will go on into lactate and hydrogen used in the Cori cycle, and then the other one is broken down into acetyl-CoA when carbon dioxide is released, and it makes citric acid, which is used in the citric acid cycle. So in that case, we go through a cycle and we yield 3 NADH and 1 FADH, which will then be used in the electron transport chain, where the NADHs will go through the 3 pumps and FADHs will only go through the first one, or the second and the third one, the last two. And then they each create hydrogen, so the NADHs will create 10 hydrogens and the FADH will create 6 hydrogens, and then those are synthesized to produce ATP. And after all the rounds of bioenergetics, we should have about 32 ATP molecules that are created that ultimately give us lots of energy.